Institute. A little bit of background about myself. So I grew up in California. I went to undergrad at UC San Diego. I spent spent 7 years in Chicago at Northwestern for medical school and residency. Spent a couple years in Portland as a chief resident and hospitalist, and then I spent the last 21 years here in Seattle initially as a pulmonary fellow and then as faculty member over the past 17, 18 years. I'm a physician scientist. I study the airway epithelium in asthma. The first few years as a faculty member, I was really interested in biomarkers, particularly exhaled nitric oxide. But then in about 2007, 2008, I became very interested in the epithelium and launched a open-ended IRB, actually now a couple of IRBs, where we collect airway epithelium from children with and without asthma who are going under anesthesia for elective procedures, and we collect bronchial brushings and nasal brushings. We collect some blood for phenotyping, and then all of those patients come back and get to pulmonary function testing and careful characterization of their clinical history. And then the cells, we bring them back to my lab and we grow them up, and then we differentiate them using the air-liquid interface model system. So we get really nice organotypic cultures. And really my lab is focused in a couple of major areas. One is the role of the epithelium in airway remodeling. And then over the past 5 or 6 years, particularly interested in innate immune responses to viruses by both asthmatic and healthy epithelial cells. So with that background, I'm going to be talking today about heterogeneous interferon responses to viral infection by the asthmatic epithelium, and then a little bit on how that relates to clinical characteristics of asthma. So I have no commercial interest to disclose. These are my funding sources. I have funding from NHLBI and NIAID. And you'll note down here, this U19 is, I run the Pediatric Airway Epithelial Core for a U19 that's led by Steve Ziegler at Benaroya. So our objectives today, we're going to try to understand more about virus-specific and host-specific interferon responses by the epithelium to viral infection, review interferon responses by asthmatic epithelium and potential associations with clinical characteristics, particularly exacerbations in lung function, review the heterogeneity of those epithelial interferon responses to viruses, and then touch a little bit on potential therapeutic interventions to modify those innate immune responses. And then if we have time, a little bit on COVID. So the main questions that I'm going to try to address are asthmatic airway epithelial type 1 and type 3 interferon responses to viruses normal, deficient, excessive, or heterogeneous like asthma itself? Are epithelial interferon responses virus specific? Are those responses associated with clinical characteristics in asthma or endotypes? What role does genetics potentially play in those responses? And then, as I talked about before, alluded to before, maybe some therapeutic interventions. So first, let's talk about viruses associated with asthma exacerbations. Now, this is a big literature. I'm just going to hit a couple of key studies, and I'm not really going to talk about natural history studies looking at viruses and their impact on the development of asthma. This is just how viruses are associated with acute exacerbations. So one of the first sort of modern studies using PCR methods was published by Gerald Teague's group in Atlanta, at least among kids in 2007 in Jackie. And it was a fairly small study looking at 65 kids with asthma exacerbations and 77 well-controlled asthmatics. They had a fairly broad PCR panel at that time, although probably not very sensitive for a number of the viruses. And really what they found was what we all know quite well today, that rhinovirus was the biggest player. Really, it was the only virus they found associated with acute asthma exacerbations, although this was probably also affected by the fact that for many of the other viruses on their panel, it wasn't very sensitive at that time. Fast forwarding about 3 years to this paper, published in Chest in 2010 from a group in Hong Kong, using perhaps slightly more advanced PCR methods. They again found that rhinovirus was strongly associated with acute asthma exacerbations. I point out here the common human coronavirus, alpha coronavirus 229E, which we see in a lot of our panels commonly, not obviously COVID. They did start to pick up that virus and a number of other viruses that we typically think of as potential triggers, but really it was rhinovirus that was the prime driver here. Then moving on to a larger study from a Chinese group just published about 2 years ago. This is an interesting study. This is a systemic review of all studies that were published up until 2017 from around the world. It includes 63 studies looking at 11 different viruses, all from studies that looked at acute exacerbations in kids and adults with asthma. So depending on the virus, the sample size varied from 344 to nearly 7,000. As you can see, pooled for both adults and kids, rhinovirus, again, was by far the biggest player, but RSV also was important. They were able to look at different regions of the world. They looked at, their studies included samples from both the upper and lower airway, but here I'm just gonna highlight the differences between the studies from children and studies from adults. You can see that for rhinovirus, again, rhinovirus and RSV are bigger players in kids as far as triggering asthma exacerbations but obviously rhinovirus is important in adults. When you look at the adults though, there's a little more balance between the different viruses. Coronaviruses, influenza, and parainfluenza virus also play an important role in adult exacerbations. And this is just graphically showing the same data, again, for kids on the bottom and adults on the top. For kids, rhinovirus is by far the biggest player, but in the adult studies, there was a little more overlap with influenza, parainfluenza, and coronavirus. What about virus-specific effects on and interferon responses by the airway epithelium? Well, this is a really interesting study published just a couple years ago from a group in Switzerland. And they were using an air-liquid interface differentiated culture model like we use in my lab. But what they did here is they actually, I believe it's about 12 different subjects. They pooled their airway epithelial cells for these cultures so that they could try to take some of the host-specific variation out of their experiments. So each transwell, differentiated airway epithelial cells, was made up of a mixture of many donors. And then they used the same MOI or viral exposure dose and then looked over a 5-day period at replication of virus. And this is all on a log base 10 scale here on the y-axis. So actually each increment is a fairly large difference in viral replication. And what they see here, or what they found was enterovirus D68, which many of you know, if you look at a lot of the viral panels we use clinically, it comes back as entero/rhino. So this is actually a fairly big player. And they found that enterovirus D68 had by far the greatest viral proliferation. The rhinoviruses, they looked at multiple different rhinovirus strains, A, C, and then rhinovirus B. They also looked at RSV and coronavirus. And the rhinoviruses had a fairly wide spread. Influenza H3N2 is here in black. One of the really striking things they showed was variation in cytopathology between these different viruses, with enterovirus 68 and influenza having a markedly greater effect on cytopathology. And this slide here demonstrates some beautiful imaging they did, confocal imaging, demonstrating this. So up in the upper left corner, you've got their control confocal microscopy, green identifying ciliated cells, blue DAPI for nuclei, and then in the infections, red indicates virus. You can see here for rhinovirus, several of the different rhinoviruses and RSV, there is some cytopathology with a reduction in ciliated cells, but it's very striking for enterovirus D68, even very early on at 2 days, as well as at 5 days for influenza. So a lot of cytopathology. So really this study just emphasizes that there are some pretty significant differences among different viruses and their effect on the epithelium. So this next study is from Larry Anderson's group at Emory. In this study, they looked at how human rhinovirus, different strains of human rhinovirus had different effects on epithelial cells. Here they used a cell line from adenocarcinoma, so it may not be completely relevant, but they looked at a variety of different cytokines and chemokines. I just really want to highlight IP10 here, which is CXCL10, which in the context of the epithelium is an ISG, interferon stimulated gene downstream of type 1 and particularly type 3 interferon. You can see this is control on the bottom. This is protein secretion into the supernatant. But between these different rhinovirus strains, there's actually a fair amount of variability in how much CXCL10 it induces. Particularly rhinovirus 14 and 16 had the greatest effect. As far as viral replication, they also found quite a bit of variability, again, log scale on the y-axis, between these different rhinovirus strains as far as proliferation of virus. This is, I think, the only murine study I'm going to show. This is a group that was looking in murine airway epithelial cells, or I think it's really lung epithelial cells. They looked at 3 different viruses that induce different degrees of disease. They used rhinovirus 1B, which is the only rhinovirus that will infect a mouse, and it doesn't really cause very much clinical disease in the mouse. They used MHV. This is a murine coronavirus, which in vivo creates a moderate disease. And then they used PR8, which is a murine influenza virus that induces pretty significant pathology. And what they found here is that there were clearly some overlap as far as genes that were upregulated and downregulated, but there's actually quite a bit more variability where virus-specific upregulation or downregulation of genes. So there's quite a bit of virus-specific effect. And this is just showing, it looked at a variety of genes, antiviral, viral detection, immune response, interferon signaling, MHC class 1. And again, you can just see by the patterns here that there's quite a bit of virus-specific effect on gene expression, at least in this murine model. What about data that we've generated? So most of the work in my lab, we've looked either at RSV or at influenza, but we do have a little bit of data, and we're building on this, comparing using the same MOI of rhinovirus 16 versus RSV in our air-liquid interface system. So these are all well-differentiated cultures. And we see pretty similar responses between the 2 viruses for interferon beta, but for interferon lambdas, the type 3, there does seem to be a little more vigorous response, type 3 response to RSV than to rhinovirus. For some of the ISGs, IFHI2, similar CXCL10, which is more downstream of type 3 interferon, a little bit more robust induction by RSV. What about host-specific interferon responses by the epithelium to viral infection? So there's very little in this area. We're very interested in this in my lab. This is an interesting study just published last year by Raymond Donnelly's group, where they took 2 different donors, and they did use an air-liquid interface differentiated model system. They also looked at 2 different strains of influenza. And what you can see here on the panel, these panels on the left are looking at H1N1, and on the right, it's H3N2, and blue is donor 1, and red is donor 2. And you can see quite different innate immune responses, type 1 and type 3 interferon responses between these 2 donors to influenza. They also looked at ISGs downstream, also differences, but what I think is kind of striking here and interesting is that for some of the ISGs, it seems to mirror the pattern that they saw at type 1 and type 3 interferon induction, whereas others, not so much. So this illustrates that not only at the level of type 1 and type 3 interferons is there heterogeneity, but probably also downstream. These are complicated pathways, and there is heterogeneity downstream of interferons as well. So this is data from my lab. We're illustrating 18 different children where we took nasal airway epithelial cells, differentiated them at an air-liquid interface, and infected them with rhinovirus-16, all at the same MOI, and then looked at interferon beta as well as some of the sentinel ISGs downstream at 48 hours. And this is all, again, on a log scale. What you can see here is a striking heterogeneity of type 1 interferon response and the ISGs downstream between these 18 different asthmatic donors. And then when we look at viral replication in those same cultures, also pretty striking log scale here, difference in the viral replication between these different donors. So what about this story of deficient epithelial interferon responses by asthmatic epithelial cells? So we're going to go back to 2005. This was a paper that most of you are probably familiar with, a sentinel paper by Donna Davies and Stephen Holgate from the UK, where they took a fairly small number of patients. They were not using and have never published any air-liquid interface studies. These are all submerged cultures, but they are primary airway epithelial cells. And they infected them with rhinovirus-16 and compared asthmatic to healthy epithelial responses. The transcript level on the left, protein for panel B. And what they demonstrated here was a strikingly lower interferon beta responses to rhinovirus infection by the asthmatic epithelium in gray versus the healthy donors. Now, in this paper, they did not tell us anything about the clinical characteristics of those donors on the asthmatic side. They also showed that the asthmatic epithelial cells, there was quite a bit more viral replication than in the healthy. A year later, they published a follow-up study in Nature Medicine. Probably the same donors, we're not quite sure. But they looked here at type 3 interferon, so the interferon lambdas, also known as IL-28A and IL-28B, and again showed what appeared to be a deficient response to rhinovirus-16 by the asthmatic airway epithelial cells in a submerged state, and a correlation, inverse correlation with viral replication. However, a little bit later, back in 2014, this is an elegant paper by Michael Holtzman's group where they looked at RSV and influenza in bronchial epithelial cells from asthmatic children. And they did not look at rhinovirus here. This is also submerged cultures. This is a little more complicated. I'll try to walk you through it quickly. Essentially, on the bottom of these are just UV inactivated virus, and then in red, asthmatic. On the left here, these are influenza infection. On the right, RSV infection. And you can see across the board pretty similar responses for type 1 interferon and type 3 interferon between the asthmatic epithelium, both to influenza and to RSV. So they postulated that there really wasn't a deficient response, at least in children, to these two viruses by the asthmatic epithelium. This is a follow-up paper published the same year by Sebastian Johnson's group, again from the UK, where they said, well, wait a second here. If we look at well-controlled asthmatics and their epithelial responses, we really don't see a difference between normal or healthy and asthmatic epithelium with regards to rhinovirus infection for type 1, and here I'm just showing type 3 interferon. So they argued in this paper that, well, there may be a deficiency in some asthmatics, but not in well-controlled asthmatics. There hasn't been a lot on that subject in the past 5 years or so. I'm going to toggle here a little bit to associations between interferon responses by the asthmatic epithelium and clinical features, particularly lung function and exacerbations. Our group is one of the few that's really looked at this so far, although there are others that are interested. This is a study that we published a couple of years ago where we used bronchial epithelial cells from asthmatic and healthy children that were differentiated at an ALI, and then we infected those cells with 2 different strains of RSV in parallel experiments. We isolated RNA, and we performed RNA seq to look at the transcriptome, and we also looked at protein in the supernatant. And we had Matt Altman help us with the analysis of this transcriptome data. And in this study, we really looked at lung function. So we dichotomized these asthmatic donors based on whether they had airway obstruction at baseline versus no airway obstruction. And really what we found here was pretty interesting. The patients, the donors, asthmatic children with airway obstruction had a more vigorous type 1 and type 3 interferon response, as well as ISGs downstream of those interferons, as compared to the asthmatic donors with normal lung function at baseline. And then Matt created a sort of geometric mean expression that sort of combined the whole type 1 and type 3 interferon and ISG signature into one metric, and found that for both strains of RSV that we used here, those donors with airway obstruction at baseline had a markedly elevated interferon signature as compared to the asthmatic children with no airway obstruction, which were actually fairly similar to the healthy controls. We also showed an inverse correlation between that signature and lung function. So we initially postulated in that paper that perhaps this was a sort of non-specific collateral damage mechanism whereby obviously interferon is very important to fight off virus as your initial innate response, but if you have too much of it, perhaps that results in airway damage. There is one study in the literature from David Proud's group in Calgary that pointed to perhaps a more specific mechanism, CXCL10, a cytokine that is, or chemokine that is part of the ISG signature downstream of type 1 and type 3 interferon. They exposed lung fibroblasts to different concentrations of CXCL10 and found that it promoted fibroblast migration. We've done a little follow-up work in my lab where we exposed lung fibroblasts to CXCL10 and found that it did increase alpha SMA expression by those fibroblasts, which is a marker of myofibroblast or fibroblast to myofibroblast transition, and also increased collagen expression. So perhaps some of these ISGs directly influence fibroblasts in the airway. What about rhinovirus? So we've done some work trying to also look at any correlations between donor lung function and interferon responses to rhinovirus. This is just showing a fairly small group of 6 donors with airway obstruction and 6 with normal lung function, although we have about double those numbers now. And we really so far have not seen the pattern that we've seen with RSV here. So we haven't seen a clear relationship between donor lung function and interferon response in asthmatics to rhinovirus. I think a lot of us kind of assume that chronic airway obstruction is linked to a history of exacerbations, and that may not be completely true for kids at least. We looked here at 70 of our asthmatic donors. I looked at their history of severe exacerbations and lung function. It really didn't find any correlation between a history of severe exacerbations and lower lung function. They're clearly outliers, but as a group, there's not really a difference. So more to follow on that. How about a relationship between interferon responses to viral infection and exacerbations? Here, the story is a little more interesting, and we're really working hard in this area. If we look at interferon responses by donors who have a history of severe exacerbation, it does appear that their interferon responses are lower ex vivo to rhinovirus than those with no history of asthma exacerbations, which seem to be similar to controls. So we're actively trying to build those numbers going forward. So what about heterogeneous epithelial responses among asthmatic patients? So I'm going to touch on this in a couple of areas. This is an interesting study published last year from the UBioPred group in the Netherlands, where they, this is an in vivo human study of adults with asthma, mild, well-controlled asthma, where they did nasal brushings and nasal washings in a run-in period, and they challenged those patients with rhinovirus 16 and then looked at nasal brushings and nasal washings at several points afterwards. I'm not going to go through all of the aspects of this study, but I want to point out particularly their interferon responses. So on the upper left here is Z-score of their interferon response, a sort of compilation of genes and the signature. And you can see that at day 3 and at day 6, there's quite a bit of heterogeneity between the subjects with regards to their interferon response. And down here on the bottom of this slide is looking at their viral load at different points. And really, ultimately, to try to make sense of this heterogeneity, they tried to break these patients down into those that had early resolution, where they couldn't detect virus by day 10, and then those that had sort of slower resolution but ultimately did clear the virus, and then those patients that didn't resolve at all. Now, this is probably underpowered, but they did notice some interesting trends. So for those that were early or late resolvers, they seem to have at baseline a little bit lower interferon responses, but then increased by day 3. Whereas those that didn't, I'm going to go back here, those that did not resolve in blue really seemed to have very little change overall during the pattern. So probably overall a less vigorous interferon response was correlated with lack of resolution. So this is just highlighting again data from our lab, the heterogeneity that we see to rhinovirus infection with regards to the interferon response, as well as replication. So is that heterogeneity in epithelial type 1 and type 3 response to viruses explained by differences in asthma endotype, genotype, or both? Well, this is an area there's not a lot of data in the literature. There's one study really from a collaboration between Johnston's group in the UK and a group out of Brazil, where they looked at sputum from adult asthmatics and sort of divided these patients into those that had an eosinophilic sputum characterization, those with more of a neutrophilic pattern, and then a mixed picture. And they argued that in the eosinophilic asthmatics and the mixed picture asthmatics, there was less interferon response, or less interferon in the sputum than in those that had a neutrophilic response. Not entirely sure what that means yet, but it's interesting. So it does suggest that perhaps different asthma endotypes might correlate with a variation of interferon responses. Obviously, Matt Altman's been looking at these kind of issues in very elegant studies in the context of the intercity asthma group, and maybe he's spoken to you more about this, but this is an active area of investigation. So what about the effects of biologic therapies on interferon responses to viral infection in asthmatic patients? Well, you're all familiar with this landmark trial of omalizumab or Xolair anti-IgE therapy in asthma. We've been using this for a very long time. One of the things that's kind of striking, we all know that for these biologics, their biggest effect is in reducing exacerbations. On the right here, you can see this interesting seasonal pattern where exacerbations peak in the spring, which is probably driven by allergies. And then there's this September epidemic or rhinovirus-triggered epidemic, particularly among kids, that seems to peak in October. And in treatment with Xolair, there was a marked blunting of both of these peaks. One of the things that's interesting in post-hoc analysis of data from this trial, it looked at, excuse me, they looked at the peripheral blood monocytes from a subset, a fairly large subset of these subjects, that they then incubated with ex vivo with rhinovirus, with or without IgE cross-linking to stimulate allergen activation. And then they looked at interferon alpha levels. And what they found was pretty interesting. So with cross-linking, there was a marked decrease in interferon expression. But then, and this is pre-randomization to Xolair, after treatment with Xolair, however, that some of that interferon response was restored. And then when they dichotomized the population based on their exacerbation history and whether their interferon response was less than the median or greater than the median, they found that those subjects with a lower interferon response had a much higher exacerbation rate than in those with a more vigorous interferon response. So essentially, they they suggested that there may be a mechanism at play whereby blocking IgE and preventing cross-linking may actually improve interferon response to viral infection. Now, what about interleukin-5 and interleukin-4 and 13 therapies? Well, there is some data. It's not quite as well developed as the anti-IgE story, but with regards to eosinophilic inflammation, eosinophils, one of their major products is a major basic protein and TGF-beta, and both of those are thought to decrease interferon lambda expression by the epithelium. So perhaps there is some boost of innate interferon response when we dampen eosinophilic inflammation. It's also, this is a nice sort of schematic from an in-press review that Matt Altman is the lead author on, suggesting, looking at the literature and suggesting that IL-4 and IL-13 may blunt type 1 and type 3 interferon responses by the epithelium. So I think there's more to come on this, but obviously all of these biologics, their biggest effect is in blunting exacerbation. So it is interesting to investigate whether one of their mechanisms is by altering or boosting interferon responses. So what role does genetics play in epithelial interferon responses to viruses? Well, to date, GWAS studies have identified 15 to 20 asthma risk loci. However, the contributions of these individual variants to asthma risk are overall quite small, or individually are quite small. And the combined effect of these loci account for very little of the overall genetic risk for asthma, which we know from twin studies is between 40 and 50%. And really, there's a paucity of data in the literature looking with regards to asthma GWAS studies, looking at asthma outcomes and phenotypic data, with one exception being polymorphisms in CDHR3, which is the receptor for rhinovirus C, which is associated with an increased risk of asthma exacerbation in young children. So we became interested in a viral sensor, MDA5, coded by IFHI1, when we looked at some of our transcriptome data from the RSV infection studies and found that two SNPs, the minor alleles, which result in a loss of function of this viral sensor, were associated with, in blue here, strikingly lower expression of type 1 interferons and ISGs downstream. So a little more about IFHI1. It codes for MDA5, which is a RIG-I-like pattern recognition receptor and RNA viral sensor. In addition to several classic interferonopathies, several additional rare IFHI1 loss of function mutations have been associated with life-threatening viral infections in young children. But there are common polymorphisms in IFHI1 that have been associated with the gain of function or major alleles have been associated with a modest increased risk of several autoimmune diseases, including lupus and type 1 diabetes and rheumatoid arthritis. So interestingly, these two SNPs are fairly common, particularly the second SNP here. The major allele is present in the majority of the population, and about 50 to 60%, depending on the population you look at, are homozygous for the major allele. So much of the literature with regards to these two polymorphisms are looking at the increased risk of these major alleles, or that these major alleles present an increased risk of autoimmunity. So teleologically, the question that we've asked is, could this viral sensor be important and a gain of function be important in protecting children from viral infection early in life that results in a selection for these major alleles, despite the fact that later in life there is a modest increased risk of autoimmunity. And the odds ratio for autoimmunity for these major alleles is is modest. It's about 1.15 to 1.3, depending on the study. So we've looked at both of these SNPs in our data set. This is looking at 51 asthmatic children that we've done these airway epithelial studies in. And for this first SNP, the homozygous genotypes for the minor allele, those donors had a 70% of them had a history of severe exacerbation, whereas those that were either homozygous or heterozygous for the major allele had only 45%. 45% of them had a history of severe exacerbation. The more common of these polymorphisms, the homozygous genotypes for the minor allele or loss of function, about 80% of them had a history of severe exacerbation, whereas the heterozygotes or homozygotes for the major allele or gain of function had a much lower rate or history of severe exacerbation. So we've been looking at this ex vivo in a lot of detail in the last year and a half. And so this is looking at ex vivo experiments where we compared those with the IFHI1 gain of function genotypes versus those with loss of function genotypes and heterozygotes for these polymorphisms, or in this case, the more common RS1990760. And we see that the viral copy number in these ALI cultures, so these are all differentiated cells that then are infected with rhinovirus 16. And then we look at viral replication and interferon responses at 96 hours after infection. And we see that the viral replication is quite a bit lower in those with the IFHI1 gain of function polymorphisms as compared to the loss of function or even the heterozygotes. And then as far as their interferon response, it's interesting. There's a much reduced interferon type 1 interferon response and ISGs downstream in those with the IFHI1 loss of function polymorphism. So we're really trying to explore this in much more mechanistic detail using CRISPR-Cas9 gene editing techniques. I'm not going to go through this whole slide here, but this is just demonstrating that over the past year in collaboration with others in David Rawlings' group and Rich James, one of my collaborators at SCRI, we have made a lot of progress in using CRISPR-Cas9 gene editing techniques to not only knock out and knock in whole genes, but also we're very close to being able to, at a high efficiency, edit base pairs of genes. So what we'd like to do is take, in the case of IFHI1, edit from a gain of function to a loss of function genotype and see what the effect is vis-a-vis viral replication and interferon responses. So more to follow on that story. So what about potential therapeutic interventions to modify innate immune responses of the epithelium beyond perhaps the biologics? Well, I think most in this audience are probably familiar with the interferon beta, inhaled interferon beta trial that was published in the Blue Journal about 6 years ago now. This was probably an underpowered study. They had, I believe, about 75 patients in placebo and 75 that were randomized to receive inhaled interferon at the first sign of a cold and then continue for 14 days. Their primary endpoint was asthma symptoms caused by respiratory viruses. It was really underpowered to look at moderate or severe exacerbations, but their primary endpoint they did not meet. There was no significant effect. They did find an enhanced morning peak flow recovery rate in the interferon group and reduced need for additional treatment. And they found a boosted innate immunity response in blood and sputum biomarkers. And in an exploratory analysis of a subset, about half in each group, they found that there was improvement in the asthma control questionnaire in the interferon group versus placebo. This initially dampened enthusiasm, although I think more recently there's been renewed interest in this, particularly looking at maybe that study was not only underpowered, but the how it's given and the timing of it, and whether it should be a continuous therapy during viral season. These are all open questions. I'm going to shift gears to azithromycin for a minute. This is something I've been very interested in over the past year. Most of you are probably familiar with this Australian trial that was quite striking. They enrolled 213 patients were randomized to azithromycin for 1 year, taken Q Monday, Wednesday, Friday versus placebo. This was add-on therapy on top of, these were all patients with suboptimally controlled asthma despite ICS-LABA therapy. And what they found was quite striking. They found a marked reduction in exacerbation incidence in the azithromycin group versus the placebo group. Now, I think their initial hypothesis and what they talk about in the discussion of this paper was that they assumed that the effect was anti-inflammatory or would be anti-inflammatory, and that would result in improved asthma control. Interestingly, though, when they looked at sputum from these patients, they really didn't see a strong signal or really any significant signal to suggest that there was a decrease in inflammation, particularly with eosinophil counts and lymphocyte counts, macrophage counts, neutrophil counts. They also looked at a little bit of a concerning safety signal. Obviously, treating patients long-term with azithromycin raises the concern of bacterial resistance. And this is a fairly small subset of patients they looked at, but they did see an increase in azithromycin-resistant pathogens in this subset of the treatment arm, which raises some concerns. So they don't talk about it in that study, but we became very interested in, could azithromycin be acting by boosting innate immune responses? And there are a couple of very small studies from the Netherlands that have looked at the effect of azithromycin on airway epithelial interferon responses, and that piqued my interest in why this might be a potential mechanism to pursue. So we have, over the past year, looked at azithromycin in our model system, and this is looking at IFHI1 response in, we decided to look at extremes of phenotype to start with. So we looked at those IFHI1 SNPs that were loss of function genotypes, and we took those patients and their grew up their cells, differentiated them, and exposed them to azithromycin. And we found a fairly robust increase in IFHI1 expression with azithromycin treatment. We also infected them with rhinovirus and used a combination of azithromycin and rhinovirus. Now, one downside of this that we're trying to pursue further is that these were concurrent exposures. So we started treatment with azithromycin at the same time as the rhinovirus infection. We're doing studies now where we pre-treat those cells for about a week before infection, and that data is not ready yet. But downstream of IFHI1, the interferon response, for type 1 interferon, we found a fairly robust increase with azithromycin. These are the loss of function genotypes, so there's not much induction of interferon beta to rhinovirus 16. There's a modest boost, it looks like, with azithromycin concurrent treatment. Similar pattern for interferon lambda, and then for a couple of the sentinel ISGs downstream. Again, the azithromycin results in a boost without viral infection, and perhaps a slight increase with concurrent exposure in the context of rhinovirus 16 infection. Okay, so I feel like over the past weeks, we've all been really caught up in this flurry of COVID-19 and SARS-CoV-2. So I thought I would try, if we have time, and it looks like we do have some time, to talk a little bit about SARS-CoV-2 and COVID-19, the disease it causes. So because of the unique resources my lab has with regards to a large number of primary airway epithelial cells and our experience with air-liquid interface model and viral infection, we've really jumped in full force in this over the past week. This is in collaboration with multiple overlapping collaborative groups here in Seattle and beyond, but we are actually starting 2 different projects right now to try to contribute to this worldwide investigation of how to defeat SARS-CoV-2. So a couple of interesting things. There's obviously not a lot published in the literature. This is one rapidly published report from Wuhan, China, where they looked at clinical characteristics of 140 patients that were infected with SARS-CoV-2. And what's interesting here, some of this you've all heard, I'm sure by now, age is the biggest risk factor for COVID-19 in patients infected with SARS-CoV-2. Interestingly, I just want to point out at the bottom here, they did try to look at asthma and allergic rhinitis, food allergy, atopic dermatitis, and they really found no patients with those conditions out of these 140 patients with moderate or severe COVID-19 in Wuhan, China. They really found no patients with those conditions, which is interesting. This is a relatively small study. There's a lot more to come on this. But these authors argue that the prevalence of asthma in this population should be 6 to 7%, and allergic diseases higher than that, and they really had no patients. So they argued that perhaps allergy and asthma was not a risk factor. Obviously, the CDC and WHO at this point are still calling those conditions increased risks, but that's, I think, probably at this point as much abundance of caution as anything else. So more to come on that. So what is going on at the level of the epithelium and the lung that results in such severe disease? And what are potential mechanistic reasons for why some patients, particularly older adult patients, have such severe disease? Well, one idea is around interferon responses. So common human coronaviruses, particularly the alpha coronaviruses, this is by far the most common strain, 229E, that's been around forever. This potently induces a type 1 interferon response, and its replication is susceptible to inhibition by type 1 interferon. If you administer interferon, that ameliorates the severity of symptoms in patients challenged with this common coronavirus strain, suggesting that interferon confers some protection. The highly pathogenic human coronaviruses, so really this is MERVs, the original SARS-CoV from 2002, and presumably, although not studied yet, SARS-CoV-2. These highly pathogenic strains seem to encode for viral proteins that have a capability of antagonizing viral sensing pathways. So that's interesting. They also seem to cause a lot of pathology with massive cell death that results in a cytokine storm. So there's this sort of double-edged sword where, of course, that leads to a dead end in viral replication, but with the price of severe pathology. There also seems to be a dramatic upregulation with these more severe coronavirus strains with a marked increase in IL-1 beta, and that was observed in infection models of SARS-CoV-1 of monocytes in human dendritic cells and tissue models. So there's a whole nother story that some of you may be familiar with, which is just fascinating, and that's ACE2 and the renin-angiotensin axis and how that may play a role in COVID-19. So in cell lines, angiotensin converting enzyme 2, or ACE2, has been identified as the SARS-CoV receptor. Really the spike protein on coronavirus binds to ACE2. But the renin-angiotensin system has a crucial role in severe acute lung injury, and it appears in animal models that were pursued after the original SARS epidemic or pandemic. The ACE2 receptor seemed to have a protective role in acute lung failure. So if you infect mice with SARS-CoV, you get considerably reduced ACE2 expression in the lungs, suggesting that reduced ACE2 expression might have a role in mediating severe lung pathology. And this injection of the spike protein into mice resulted in worsened acute lung failure. And then if you block the renin-angiotensin pathway with either ACE inhibitors or angiotensin receptor blockers, that reduce the pathology. So this raises this interesting question: is ACE2 expression protective or is it detrimental? Because obviously it's the viral receptor. So on the one hand, simplistically, you'd think that if there's more ACE2 expression, that's a risk factor. Well, in animals, ACE2 expression is markedly higher in younger animals. We don't have data really from kids at this point, human data from kids. But in animal models, the younger the animal, the more ACE2 expression on the epithelium. There is some adult data that in older adults, ACE2 expression decreases. So you've got this really interesting, potentially paradoxical situation where ACE2 is the viral receptor, and when the spike protein binds to ACE2, it's cleaved from the membrane. So and ACE2 is really important in antagonizing ACE and the angiotensin receptor signaling pathway. If ACE and its downstream signaling pathways are not antagonized by ACE2, you get fairly profound pulmonary edema, poor cardiac contractility, ARDS, and ACE2 seems to counter-regulate that. If you give recombinant ACE2 to mice with SARS or influenza or RSV in animal models, that actually protects them against severe pulmonary disease. So it may be one hypothesis that we're pursuing in collaboration with Lisa Frankel's group and Whitney Harrington is that in kids, perhaps there's a lot more ACE2 expression at the level of the epithelium, and that you need some ACE2. Obviously, ACE2 is a pathway in for the virus, but if you lose it with the cleavage of the ACE2 protein from the epithelial surface, then suddenly, at least later in the infection, you can't antagonize ACE1 and the renin-angiotensin system, and you end up with severe pathology. Whereas in younger patients, they can spare to lose. They can be infected with the virus, but they've got a residual ACE2 that then prevents the more severe pathology. So this is a really fascinating open question. We don't really know what the right answer is, but we're pursuing experiments right now to look at both this question of the ACE2 and the renin-angiotensin axis and how that may relate to disease in COVID. And we're comparing airway epithelial cells from both kids and adults to try to address this. And then we're also looking at interferon responses to SARS-CoV-2, dichotomizing based on our already existing knowledge around polymorphisms in IFHI1. So in conclusion, I didn't think I'd get through all these slides this quickly, but in conclusion, I would make the statement that type 1 and type 3 interferon responses to viral infection by the asthmatic epithelium is not easily dichotomized into high versus low. High epithelial interferon response to RSV in asthmatic children does seem to be associated with lower lung function. Is this virus specific? It seems to be, at least in our data set, more specific for RSV and less so for rhino. The literature is obviously mixed with regards to whether a low asthmatic epithelial interferon response to viruses is associated with exacerbations. I would argue that some of these differences between studies may be explained by differences in asthma endotype, genetics. In our case, we're very interested in viral sensing, polymorphisms, or perhaps both. The efficacy of current biologics in reducing exacerbations may be partly due to enhanced innate immunity. And then finally, therapies to enhance type 1 and type 3 interferon responses by the epithelium may prove efficacious in subgroups of patients with impaired interferon responses. So perhaps there may be a role for drugs like inhaled interferon if we can target them to the patients that really have a deficient response. So just quickly, I'll acknowledge folks in my lab that have contributed to all of this, as well as collaborations in the Rawlings lab and the James lab, particularly with our gene editing work. And then of course, collaborations with Stephen Ziegler and his group and the entire U19, and Matt Altman, who's helped tremendously with our RNA-seq analysis. And I think that's all I have at this point. So most of you have your mic. Well, actually, there's one more thing I have to do. Your word of the day is virus. I've been told by Sonia I need to show that slide. So there you go. And most of you have it muted at this point, but if you want to unmute, if you have any questions, I'm happy to try to answer those best I can. Any questions from those that want to ask them?